Let's take a look at the important terms for the sensory physiology lab. This is also labeled Lab 7. It used to be together with the muscle physiology lab in your lab manual, and then somehow, I don't know what happened to it, it kind of got lost in your lab manual, but I posted the handout for the sensory physiology lab in your Canvas folder for lab stuff, and it's also there's another link in the modules for this week. So I think sensory physiology is really important, and plus it's easy to do some of the sensory exercises at home, which you're going to have to do. So let's take a look at the important terms. We're going to start out here with sensory transduction. That always involves a receptor, and so there are different ways you can list receptors or make sort of tables or group the receptors. Easy ways to go by function, and in physiology we're into function, so let's do just that. If a receptor responds to temperature or temperature changes, we call it a thermal receptor. We have actually cold and hot receptors separately, and you can test that out on a patch of skin by having like a warm or hot blunt probe would work best. That touches your skin, and then you can figure out where do you have cold receptors on your skin, where do you have hot receptors. So that's kind of a fun way to map out a patch of skin. Chemoreceptors respond to chemicals. They'll be in your tongue, for example, or in your nose. You have lots of chemoreceptors. Also inside of our body we have chemoreceptors that measure CO2, oxygen levels, hydrogen ion levels. Then mechanoreceptors, those will be your touch pressure receptors that respond to mechanical kind of stimulation. Photoreceptors, they're only in your eye. They respond to electromagnetic energy in the visible range. Nociceptors, sometimes also called nociceptors or nocireceptors, those are pain receptors. They respond to painful stimuli. And then proprioceptors, they are giving information about body position, and you would have those in the tendons of your that are attaching your muscle to bone. Now, another way of classifying receptors would be to say whether they are phasic or tonic receptors. Phasic receptors means that they stop firing after a certain amount of time. So phasic receptors, easy way to remember that is they phase out. Think about the famous garlic room. You first enter the garlic room, smells really strong there, and then you get used to it, or your receptors stop firing because they're phasic receptors, and it feels like as if the stimulation is not there anymore. Well, it's still there, but your receptors stopped reporting because they are phasic receptors. Tonic receptors don't do that. They keep firing until the stimulus is not there anymore. So they have a constant firing rate. Typical example for that would be pain receptors. You feel the pain until the painful stimulus is not there anymore. Those are tonic receptors. Another way of classifying receptors is extero versus intero receptors. Extero receptors are on your surface, on the outside of your body. Intero receptors would be inside of your body. Sensory adaptation, it goes along with the idea of the phasic receptors. So olfactory adaptation would be that your chemoreceptors stop firing even though the smell or the scent is still there. Tactile adaptation is more of like a learning procedure that directs you to certain movements. Balance, well, here's a little exercise for you to do. Try that out. Stand on one leg for about 10-15 seconds. You should have no problem doing that because your sensory system gives multiple inputs about your body position and the balance that you can find. Your eyes are involved, so are your proprioceptors, and then also your inner ear that gives you information about your balance and body balance. Now, eliminate one of those pieces of information, and right away you will notice that you're gonna have trouble finding your balance. So same thing, stand up on one leg and now close your eyes, and try that out for about 10-15 seconds and see how just your eyes participating in you finding your balance. It's actually pretty dramatic, it's pretty drastic. So try that out, please. Just a short exercise, stand on one leg, eyes open, then stand on one leg, eyes closed, and note the difference. You can also, if you have a office chair at home, you can try rotational acceleration. So that would be on a chair. In the lab, we would do that with our lab chairs. Try to rotate yourself, or have somebody else rotate you on a chair, and see what that feels like. Linear acceleration, you can try that. Well, actually, you can try that just with your car or with a bike. Feel what that feels like. Now, a couple exercises are actually easy for you to do at home. You can test your eyes, your vision, and for that I added a Snellen chart to your lab folder for this lab. That's just checking for visual acuity. So the Snellen chart has letters of different sizes on it. You're supposed to be standing 20 feet away, and 20/20 vision means that from 20 feet away, you can see what a person with normal, perfect eyes can see from 20 feet away. And that should be, I think it's the second smallest line. So the second line from the bottom, you should be able to see that one, read that from 20 feet away. If your vision is worse than 20/20, that means that you can see from 20 feet away what somebody with perfect normal eyes can see from, let's say, 50 feet away or 200 feet away. So if you can only see the big letter E on the top, that would be not so good. So I found a more quiet spot here to record this. I apologize for the noise in the background, so hopefully you can still hear everything. But anyway, let's move on to astigmatism. Astigmatism is basically a distortion in the image that you're producing because there's an imperfection in the curvature of your lens or the retina. And astigmatism is very common. Most people have at least some form of astigmatism. You should be able to find an astigmatism chart. It's basically a symmetrical image with a circle in the center and then lines that are radiating away from that. If you see any kind of distortion in these lines, that they're not 100% parallel, or that some lines are thicker than others, or that they're somewhat bent, or that some of them are closer together, that would all indicate astigmatism, which is something most of us have, some more than others, but it is due to an imperfection in the curvature of your lens or your cornea. The blind spot is the spot in the retina where the optic nerve attaches, and because all the ganglion cells are bundling up to become the optic nerve, you don't have any room there for receptors. And if you don't have receptors, well, then you can't have a perception. So the blind spot is the spot in the retina where there are no receptors because the optic nerve is basically bundling up there and leaving the eyeball. If you're projecting an image onto the blind spot, well, then you won't see anything. You can try that out. In that lab exercise that's posted, there are some exercises where you can test yourself and try to project something onto the blind spot and watch how the image disappears and then reappears once you are projecting on a different spot in the retina. Color blindness is a genetic problem. It's not really actually a problem. It's maybe you will have bad taste when it comes to picking clothes or, I don't know, decorating your home. It's the inability to distinguish certain contrasting colors. The most common form would be red-green color blindness. So there, where you have trouble finding or distinguishing contrasting red and green colors. And in order to test for color blindness, there are these Ishihara pseudo-isochromatic diagrams that we have in our lab, but I'm sure you can find them easily online as well and test yourself if you have color blindness. Most people actually know it's a much more prevalent for males because it's actually on the X chromosome, the pigmentation or this ability to distinguish colors. And if you only have one X chromosome and it happens to be affected, well, then you have the color blindness. Women have 2 X chromosomes, and if at least one X chromosome doesn't have the issue, then you would not experience color blindness. But you should be able to figure that out. And visual illusions, I have some in the back. We'll show you some, and we can take a look at that. There's some interesting things that you might enjoy doing on your own. And the reflex, that is sort of to tie in with the muscle intro that I have on the other video. Make sure that you are taking a look at the knee-jerk reflex. I'm going to draw this one more time because I figured out how to make the pen right smaller. So the knee-jerk reflex, you would have the patella tendon right here. So this is the patella tendon. You tap the patella tendon with a reflex hammer. The sensory information will be sent. So here's the cell body of the sensory neuron. Will be sent to the, oops, I think I might be leaving the screen right now. Not too bad. So we're gonna move the, hang on. So we're going to put the sensory information into the spinal cord back here. This would be the posterior horn, here the anterior horns. It's kind of something like this. So here's our sensory input coming in. Then we're synapsing to an interneuron. I'm going to label this I. This is the sensory neuron. And then we're synapsing here with this interneuron to a multipolar motor neuron. Let's draw it like this. That's what they look like. And that will send the information back out, and then you're going to have your gastrocnemius muscle here then starts to flex. Okay, so some of my students, they like to remember the reflex arc right here. SIM, sensory intermotor neuron. That's maybe a good way to remember that. So sensory neuron, interneuron, motor neuron. The important thing is in a reflex you don't have any ascending or descending tracks. So you're going into the spinal cord and right back out, and that's how it's very fast, and you do not think about it. There's no evaluation going on. Now I skipped down a few pages toward the end of this handout. I just wanted to show you some visual illusions just to point that out. Here in this image, what you want to look for is, do you perceive these lines as curved or as straight? Well, if you think it's curved because that's what you see, well, measure and see if they really are curved. Moving down here, this is an image on perspective that you probably can't see that well. Even though these two balls are this exact same size, we perceive the one in the back as bigger because our brain makes that up. And here, this triangle, you can take a look at that. Here you can decide whether you see a face or a vase. Here you should decide, are you seeing a rabbit or a duck? This one is similar to a previous, to the triangle. Check out this one here. Stare at the center and then move your head toward your screen that you're looking at, and then away, and watch what happens. And then here, this is probably my very favorite one. Doesn't look like much of anything, but go ahead, stare at the center, that little dot in the center. So you want to stare right here at the center of this, and once you stare at the center for about 10 seconds or so, let me make this a little bit smaller so we can actually have this in the whole frame. So you're staring at the center, keep staring, and then now you want to move your head toward the screen or the paper that you're looking at and away, and watch what happens to these outer circles. Basically, it's kind of interesting. So check it out and have fun with this. Please be sure to take a look at the folder. There are several things posted. This handout, which I was just using for the front terms, and then here for the visual illusions, there is a revised version of this lab posted that is more specific with online instructions. Please take a look at that and do as many of these exercises as you possibly can at home.